Okay, this is where we left off, and now we're going to think about the idea that an electron transport chain is available and our terminal electron acceptors are also available. So if those two things are true, we can continue the breakdown of our pyruvic acid into the transition step into the tricarboxylic acid cycle. So let's look at our transition step first. We can again think about what goes in and then what comes out. What goes in is our pyruvic acid from glycolysis. What comes out of our reactions of the transition step is our acetyl-CoA. As our pyruvic acid gets broken down, we lose a carbon dioxide molecule. And then we have more electron acceptors coming in. NAD comes in, picks up electrons and hydrogens, leaves as NADH. And we have an enzyme helper molecule, a coenzyme called coenzyme A, also known as CoA, comes in, picks up the remaining molecules and brings them into the tricarboxylic acid cycle. So CoA comes in, makes acetyl-CoA, brings that molecule into the tricarboxylic acid cycle, and then coenzyme A is regenerated. It grabs a new molecule, brings it in, is regenerated, grabs a new molecule, brings it in, and is regenerated. So coenzyme A is just there to shuttle molecules into the tricarboxylic acid cycle. So acetyl-CoA is the molecule that's going to enter the TCA. So pyruvic acid, we lose a carbon dioxide. Our electron carriers come in, NAD goes in, NADH comes out. Coenzyme A attaches, becomes acetyl-CoA, and that brings our molecule into the tricarboxylic acid cycle. And then we enter the tricarboxylic acid cycle, and our molecule goes through many intermediate steps. One important thing is that carbon dioxide comes out of our tricarboxylic acid cycle. Now in a human, the carbon dioxide made in the transition step and the carbon dioxide made by the TCA, that's the stuff that we breathe out when we send this stuff to our lungs. We breathe it out of our bodies. Now, obviously, microbes and single-celled organisms, many organisms don't have lungs. This carbon dioxide will simply diffuse out of the cell. Over here, we have places for more electron carriers to come in and strip more electrons and more hydrogens away from our molecules. NAD comes in, picks up electrons and hydrogen, and leaves as NADH. Another important electron carrier comes in, FAD, picks up hydrogens and electrons, leaves as FADH2. Now let's add another part to our reaction here, because the tricarboxylic acid cycle also makes a little bit of ATP, again by this mechanism of substrate level phosphorylation. So we can represent an ADP coming in, having that third phosphate added and leaving as ATP. So again, our tricarboxylic acid cycle is able to generate 2 ATPs. So over here, we made a small amount of ATP through substrate level phosphorylation in glycolysis. And down here, we made another small number of ATPs through substrate level phosphorylation in the tricarboxylic acid cycle.